All right, so now we're going to continue with what's called translation. So this is the process where we have now a piece here of messenger RNA. And it has something called a 5' cap added to it and a poly A tail. And the 5' the poly A tail protects it in the cytoplasm. So these are things that are I put a different color because they're added after transcription. So they're not transcribed, they're not copied from the DNA. They're added afterward as part of the processing for messenger RNA. So this just kind of protects it when the messenger RNA is in the cytoplasm, and the cap is a binding site. So it's a binding site for the initiation part of this. So what's going to occur then is the small subunit and the transfer RNA with methionine, okay, are going to assemble first. This is the small subunit, SSU, of the ribosomal RNA, and I'm trying to get the color here going. Okay, so I'll have this be for the transfer RNA. Transfer RNA, and I'm going to put met here for methionine. So these things bind to this cap, okay, and then they're attached. Color consistent here for you. Bind here like this. And now the anticodon here is then reading. So ACA. Okay, but the methionine codon is going to be AUG. So we need to find that. And that's not it. That's ACA. So it moves one nucleotide at a time. So now it really goes into CAG. And that's not it. So it moves. So it's moving in this direction. So then it's like AGU, no. GUA, no. UAU, no. AUG, yes. Okay, so now once it moves along and the codon and anticodon pair up. So we're like, okay, now we found where we're going to start. So that is called the start codon, and we'll actually find that all proteins begin with methionine. And it may not stay in the final protein. It often gets cut off afterward in processing of the polypeptide. But to begin with, that's going to be where we begin. So it's like, remember, in the DNA, there was something that said start here, right? There's something in DNA that says begin. That was called a promoter. That's a DNA sequence. Usually it is a specific sequence. I didn't put this in the last lecture, but I'll put it here now just because it's going to be a little bit helpful when the next thing I talk about. A common sequence in the promoter of the gene is TATA. The sequence for the start codon is AUG. One of the ways for you to keep these separate, right, and remember them and understand them is, remember, T is only in DNA, U is in RNA, right? So AUG can't be part of the DNA because that's only in RNA. TATA can't be in RNA, it has to be in DNA because T is only in the DNA. So again, if you had that little bit of a rule, then this is helpful and makes sense. If you don't really know what that means, then it obviously isn't as helpful as it should be. So now once these things find the start codon, remember there was another subunit. There was another thing called the large subunit. Now this guy isn't here, he's moved, so I'll erase him. Is that, okay, it's moved along. And now it's moved to this new location. And now, once this happens, the large subunit comes in. This is a large ribosomal subunit. It actually has several different types of RNA and proteins together. It has these, I drew the lines here because there's binding sites. So like an enzyme has the active site, the binding site, the ribosomal RNA has active sites or binding sites, right? And so they're given particular names, right? There's an A site, a P site, and an E site. So Initially, it starts off a little bit contrary to the definition of each of these sites, but that's because this is where we're beginning the process. Typically, the A site, right now, you see it's open, and it's ready for a new transfer RNA with some amino acid on it to come in here. So what can happen now is the A site can have a transfer RNA come in. Amino acid, is it amino acid? I'll put number 2 question mark. So we would be checking, or in this particular process, they are checking codon with anticodon. So what's the anticodon do you need for CCU? In this transfer RNA, it would need to be GGA. And if this one had GGA, then that means it's correct and it would stay there. And yes, this would be amino acid number 2. And so what would happen is this enzyme, or this ribosomal subunit, would cut away methionine from this transfer RNA, and it would make a peptide bond. Remember when we studied proteins, amino acids were joined together with these peptide bonds, and so this is how they're joined together. They're joined together by the large ribosomal subunit, a specific enzyme in it called peptidyltransferase, transfers the amino acid from one to another and makes the peptide bond. And so now what you have is a transfer RNA with actually 2 amino acids attached to it, an empty transfer RNA, and then an empty site. And so now the whole thing will start to make sense, okay, because what's going to occur is this whole thing is going to move. So what I'm going to try to do here, you can actually see action like take place. Attempt to do this is to move the large subunit. Okay, so now the large subunit has moved forward. And now we have the E site, the P site, and the A site. Remember, the A site is where new transfer RNAs carrying single amino acids are going to enter, and now it's open and ready. The P site, so we have an A site, that's for transfer RNAs with new amino acids. The P site is going to contain transfer RNAs with a peptide. So now once we start to add amino acids together, that's a peptide. And as we get many of them, it'll be a polypeptide. That's going to be what we usually find in the P site, a transfer RNA with many amino acids attached to it. In the E site, usually we find empty transfer RNA. And it's also, sometimes you can use the E for exit. So it's going to be the exit site. So this empty one isn't really going to stay here very long. It's going to be kicked out. An empty transfer RNA is removed from the E site, kicked out of there. And then it can be recycled again. It'll come around, pick up another amino acid, and then eventually it can come back to the A site. But for now, it's kicked out, and the A site's ready for another transfer RNA. Is this amino acid number 3? I don't know. So what we see, so it comes in here, it checks against the codon. So it's supposed to be, if this here is AGU, we need UCA. But if it was UGC, say, wait, that doesn't write at all. Like this one pairs up, but the others don't pair at all. So if it's wrong, it just kicks it back out. Right? And so this whole thing would just be kicked out of the site. But nope, not the correct codon, so we're kicking you out. Right? So then a new one would come in. So okay, here's another one. Maybe this one is the correct one. So it comes in, it binds to the site, and now we have, UCA. Oh, that's correct. And so if that one is correct, then the whole thing will move again, right? And so the bond would be cut here. So this guy's already gone. He's not even there anymore. He's completely kicked out. Okay. This bond is broken. Amino acid 2 is detached from this transfer RNA. And then another peptide bond is made between these two, like this. Now this one's empty. He's going to go into the E site. This one has the peptide. He's going to go into the P site, and then the A site will be open because the ribosome is going to move. And so this is what's going to just keep happening over and over and over and over again. So what we've talked about so far are two stages of translation called initiation and elongation. So initiation was when a small subunit and the methionine transfer RNA attached to this cap read along and found the start codon, AUG. That's to initiate or to begin the process. Once they found it, then the large subunit came in. That ended initiation, and we start elongation. Elongation, that's where we get transfer RNAs coming into the A site, peptide bonds forming, the ribosome moves. New transfer RNA with amino acid comes in. If it's correct, peptide bond, ribosome moves. And it's just going to keep repeating the exact same thing over and over and over and over again. Eventually, what will happen is after building a long polypeptide, the ribosome will come to a place where we have all these amino acids attached to it. Yeah, let's see, how many would we have here? That's the methionine, and then 2, 3, 4, 5, 6, 7, 8, 9, 10, and then we're going to be right here. So let's say there's 10 of these here. Most proteins and polypeptides have way more than 10. Just to give you an idea. So there's a chain of amino acids here with methionine on the end. That was our first one. And now in this in the A site, we have, that's the P site here, the site was empty. This sequence, UAA. And what we're going to find is that UAA, UAG, and UGA, these three fell out, no amino acid. They're a stop sequence. So what happens, there is no transfer RNA with any of these codes. So there's no one to come into that site. Instead, something else comes into the site called a release factor. So the release factor comes in, attaches. The protein here thinks he has another amino acid, so he breaks this bond in order to form the new bond, but there's nothing there to attach it to. So that makes this polypeptide released. So the release factor triggers the release of the polypeptide. And then this empty transfer RNA just gets kicked out. The ribosome pieces actually pull apart, and then the whole process ends, and that's called termination. So during termination, we find a stop codon. Our initiation is where we looked for the start codon. Our elongation is where we just keep reading new codons after codons after codons after codons and adding amino acids to make this very long polypeptide chain. And that's called translation, sometimes also referred to as protein synthesis, because it's how cells make proteins. This happens in the cytoplasm of cells, not in the nucleus. So all these pieces of RNA are copied from the DNA in the nucleus, and then they have to leave the nucleus to go out in the cytoplasm. And that's where this happens in eukaryotes. Obviously, bacteria don't have a nucleus. So the DNA is in the cytoplasm. So when the RNA is made, it's immediately already in the cytoplasm. In our cells, it has to be transported or moved out from the nucleus to the cytoplasm, and that's where this process happens. So key takeaways from this are the stages of translation. Or first off, what is translation? It's where we use the information in RNA to make the polypeptides. What RNA? Well, we have three kinds of RNA: messenger, transfer, and ribosomal, all working together. Each of these were coded for by different genes. There was a gene for the messenger RNA. There's a gene for this transfer RNA. a gene for this transfer RNA, a gene, so there's different genes for all the there's a gene for this ribosomal RNA. There are multiple genes that code for the ribosomal RNA that's in the large subunits. There's all these genes coding for RNA. Some of those were coding for the sequence that will become the amino acid sequence. This is where they all assemble. Translation. Initiation, finding the start codon. Elongation, large subunit comes in, reads the whole thing. Termination, reads to a stop codon, and the process ends. And so that's the end of translation. And that would be that polypeptide could become a functional protein in the cell. Right? And that would be then the expression of the gene. So the gene in the DNA has now been expressed as a specific enzyme or as an active transport pump or some other thing that will do a job in the cell. So if you say genes code for eye color, technically that's not really accurate. The gene would code for a sequence of nucleotides that codes for a sequence of amino acids that creates a certain protein, and that protein is an enzyme who creates pigment, and that's what gives you that color. So the color comes from an enzyme chemical reaction. The enzyme comes from this process, from translation made by that. And then the information to make it was stored in the DNA. So it's kind of this backward, forward, DNA, RNA, protein, and how they're all related to each other. That's the gene expression process.